Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 8. In this video you're going to be learning about multiple alleles. And by the end of this video you should be able to define what multiple alleles are and describe the allelic series in genes with multiple alleles. So just a bit of background information. In Gregor Mendel's experiment on peas, he studied 7 different traits: seed shape, seed color, flower position, plant height, pod shape, and pod color. Each of these 7 traits were controlled by just 2 alleles, a dominant allele and a recessive allele. For example, for the seed shape trait, there were only 2 alleles, the dominant round allele and the recessive wrinkled allele. For the flower color trait, there were also just 2 alleles, the dominant purple allele and the recessive white allele. But in the real world, many genes have more than 2 alleles in a population. Sometimes genes can have 3, sometimes genes can have 4, or even more alleles in a population. Remember the slide from video 7, incomplete dominance and codominance? I said in video 7 that the trait for solid dark coat colors in cats is controlled by the brown gene located at the B locus of the cat's genome. That's true. But then I said that the B locus contains 2 alleles for this brown gene, one allele being the wild type black allele, capital B, and the other allele being the chocolate allele, little b. This was the lie. The brown gene located at the B locus actually has 3 possible alleles. So the third allele is B1, little b1, which is cinnamon. Cinnamon is recessive to both black and chocolate alleles. So for a cat to express the cinnamon phenotype, they must have 2 copies of the recessive little b1 allele. So going back to what I said in the previous lesson, the chocolate allele is indeed recessive to the black allele, as shown by the greater than symbol, but the chocolate allele is dominant to the cinnamon allele, as shown by the greater than symbol in this allelic series. For your information, an allelic series describes the linear hierarchy of dominance for a set of multiple alleles for a single gene. In this case, the same case we were talking about in the previous slide, black is more dominant than chocolate, which is more dominant than cinnamon. And in this diagram, there is a scale going from the most dominant to the least dominant to emphasize the linear hierarchy of dominance in this allelic series. I also want to point out how this diagram uses an underscore or a line to show the dominance of the B, the capital B allele. This underscore is saying it doesn't matter which allele is in this spot. It doesn't matter what the second allele is. It could be capital B, it could be little b, it could be B1. The phenotype will be black nonetheless because in the complete dominance pattern of inheritance, which is what we see here, only one dominant capital B allele is needed to express the dominant black phenotype. And I've included some very cute photos of cats. So the brown gene in cats is a great example of multiple alleles. Multiple alleles refers to the presence of more than 2 alleles for a gene within a population. I stress the more than 2 because too often in exams we see people write more than 3 alleles or 3 or more alleles, which is incorrect. You should say more than 2 alleles for a gene within a population. When multiple alleles are present, the inheritance patterns get a little bit more complex. That's because multiple alleles can lead to a variety of phenotypes, and the number of possible phenotypes is determined by the dominance relationships between these multiple alleles. Sometimes alleles show complete dominance, sometimes they show incomplete dominance, and sometimes they show codominance. So we use an allelic series to illustrate the relationship between these multiple alleles of a gene, like what we saw in the cat's brown gene in the previous slide. All of the 3 alleles, in this example, show a pattern of complete dominance. If you can't remember the difference between complete dominance, incomplete dominance, and codominance, this is the point where you pause the video and go back to watch video 7, which is all about the differences between complete dominance, incomplete dominance, and codominance. Let's look at another example of multiple alleles. Remember this slide from video 7? I talked about the trait of human blood types as an example of codominance. IA and IB alleles are codominant to each other, so in the heterozygote, both A antigen, IA, and B antigen, IB alleles are expressed together, equally, and independently in the phenotype. But after what I just told you about multiple alleles, what do you notice now? Do you see it? Can you spot the third allele? The third allele is the little I, which codes for no antigen and produces a phenotype of type O blood. The lowercase I tells us it's a recessive allele, meaning 2 copies of this allele are needed to produce the phenotype of type O blood. So the trait of blood type in humans is controlled by the ABO gene on chromosome 9, which has 3 genetic variants, or 3 alleles. The recessive I allele that produces a non-functioning protein, the dominant IA allele, which produces an enzyme that forms the A antigen, and the dominant IB allele, which produces a different enzyme which forms the B antigen. And the way these 3 alleles relate to each other is represented by this allelic series. IA and IB are codominant, hence the equal sign. Both alleles are expressed together equally and independently in the phenotype. When both alleles are present in the genotype, so when the genotype is a heterozygous AB, both A and B antigens are expressed in the phenotype, hence the heterozygous individual has type AB blood. Both alleles are dominant over and mask the recessive little I allele, hence the greater than sign. So when the A allele is present with the O allele in the heterozygous AO genotype, the dominant A allele masks the recessive O allele, and the heterozygous individual has type A blood. The same goes for when the B allele is present with the O allele in the heterozygous BO genotype, the dominant B allele masks the recessive O allele, and the heterozygous individual has type B blood. Because the O allele is autosomal recessive, 2 recessive O alleles must be present in the genotype in order to be expressed in the phenotype as type O blood. In this last example, you're going to see how within the same allelic series, there could be complete dominance interactions and incomplete dominance interactions too. Here we're looking at the trait of color point restriction controlled by the C gene in cats, where there are 4 alleles present in the population. The source specifically notes that the black to white gradient here in this schematic represents the amount of pigmentation, not the color of the pigment. The first allele in this gene is the wild type big C allele, which is expressed as a fully pigmented, full-colored coat. The second allele is the Burmese allele, which is CB, expressed as the Burmese color point restriction phenotype, where the extremities are pigmented and the body is only slightly lighter than the extremities. The third allele is the Siamese allele CS, which is expressed as the Siamese color point restriction phenotype, where the extremities are pigmented but the body is very light or white. And finally, the fourth allele is the albino or the colorless allele little c, which is expressed as a completely white coat, totally lacking of any pigmentation, not even in the extremities. The allelic series above shows us the linear hierarchy of dominance for the multiple alleles of this gene in this population. The wild-type big C allele is dominant over all the other alleles, as shown by its leftmost position and the greater than sign. The Burmese allele, CB, is incompletely dominant over the Siamese allele, CS, as shown by the equal sign. And both Burmese and Siamese alleles are dominant over the little c albino allele, as shown by the greater than sign. So as you can see, there are 4 alleles in this allelic series, yet there are 5 possible phenotypes. That's because the incomplete dominance pattern of inheritance produces a 5th phenotype in the CBCS heterozygote. CBCS, right here. As I discussed in video 7, the Burmese and Siamese alleles are blended to produce a new mink color point restriction phenotype, where the body color is not as dark but not as light as the original alleles. Well done, you've reached the end of the video. So by now you should be able to define multiple alleles and describe the allelic series in genes with multiple alleles. Thanks for watching, and I'll see you in the next video.